รู้ไหม
ปิดประตูด้วย
Wari, I just have like a quick question. How long are we gonna be in the session? Do you know if we're gonna take like an hour?
Yeah,
Or
but if you
2
have
hours?
to leave first, then I can send a recording to you.
Oh, okay. Yeah, sounds good.
Okay. So, do you guys have any pen, paper, or anything that you want to take notes with? You can take it out. And then we're going to go over the question together. Have you guys finished all the content in the lecture?
No, she's still like she barely started on chapter 10 on Wednesday.
Okay, what is chapter 10 again?
Chapter 10 is sorry, give me a second. Chapter 10 is photosynthesis.
Okay. So, any question about photosynthesis? I guess you can take notes, and once you finish, you can try do it on your own. All right, and I have an answer for it. So yeah. So let me review all the things that we're gonna go over in exam 2. We're going over about membrane permeability, right? Like osmosis, things like that. You guys learn that?
Yes.
Okay. And have you guys finished cellular respiration?
Yeah, we finished it.
Okay, you guys finished that, and you have photosynthesis left?
Yes.
Okay. So, okay, are you guys ready? I'm gonna start now.
I was born ready.
You was born ready? Yes, we love that energy. All right, keep it coming. For exam 2, I have. Oh, okay. Okay. So this is general picture of about photosynthesis. So you're supposed to know more in detail for these guys. Just know in every detail and also in big picture too. And also cellular respiration. You see this. Remember it and also go deep in details too. And remember numbers of ATP too. That's very important. Okay, let's go into our first question. Channel and carrier protein that only diffuse mechanism to move solute through their pore opening are said to be involved in A. Passive transport, B. Active transport, C. Co-transport, D. Countertransport, C and D. I'll give you some time to think about it. And then you can tell me when you're done or do you want to go over it together, okay? Samantha, you're done?
Yeah, I wanted to say something, but I was hesitant. But is it A?
Passive transport. So can you walk me through why do you answer that one? How do you come to this answer?
Okay, because like cannot carry personality diffuse mechanism. Oh, because isn't diffusion a part of passive transport? That's how I kind of remembered it.
Mm-hmm.
Well, that's why I thought it was passive transport instead of active transport.
Mm-hmm. Samara, do you have the answer yet?
I'm gonna go with B, active transport, because they're actively moving.
Mm. What does active mean when you, in biology? What does active mean?
Worry, I was gonna say, I don't know if this is gonna make sense, but
Mm-hmm.
how I would remember it, like active is going up the concentration gradient and passive is going down the concentration gradient. So that's
Mm-hmm.
why I said it was passive.
Mm-hmm. Active is against concentration gradient. Because it's against concentration gradient, you use ATP, right? So when we say active, it's mean we're using ATP to go from low to high. As Samantha said, passive means no ATP and it cross the membrane high to low. Okay. So what can you tell about diffusion? Does diffusion use energy or no?
I hope I'm right, but is it no?
Yes, no.
Because it's going across the membrane?
Mm-hmm. Across the membrane in high to low, right?
Yes.
Mm. So I think we are right. Okay, so Samara, do you understand how I explain it to you?
Yes.
Okay. So wait. Ah, no, come back. I already give you. Okay, so the right answer is passive transport. Yay! Now clear. Let's go over next one. Active transport across membrane is facilitated by carrier protein, ATPase pump, co-transport protein, counter-transport, all of the above. So what can you tell right from the get-go from the question? Active transport. What does it mean?
It's going up the concentration gradient and technically it has energy.
Yes, it's use energy and against concentration gradient. Now we have to come back and look at A, B, C, D, E. You have to look at all those choices and compare which types of protein use ATP. So this one, I would say, um, it's all memorization. It's not like adapting your knowledge yet. But yeah, it requires you to recall what is active transport is and what type of protein does use ATP. Okay, I'm gonna give you guys a time and to think about it and let me know when you're done.
Worry. Can I say the answer?
Mm-hmm.
Or I don't know. I forgot about this one, but I think is it B?
Samara, what's have you come up with your answer yet?
Not yet.
Not yet. Okay. Why do you answer B?
Well, since it's asking since active transport has ATP, I thought of it from like the hint right there where it says ATP in
Mm-hmm.
it. That's how I thought of it.
Mm-hmm.
You know what? Isn't it okay? You know what? I think I got mixed up. I think isn't it like all of the above?
Yes. Okay. So how do you come to all of the above?
Because active transport also has co-transport proteins and counter-transport proteins, but it also has carrier proteins and ATP pumps.
Mm-hmm.
Because active transport has energy, and energy is required for all of these.
Mm-hmm. Yes.
And
So carrier protein can either be passive or active, all right? It can either use ATP or not use ATP. But the other thing else, ATP is pump, co-transport protein, counter-transport protein, all of these are strictly one that use ATP. So that's how our answer came to be all of the above. Okay. Samara, do you want to ask any question? Are you confused on anything?
No, I'm just looking to the notes.
Okay. Is it okay if I go to the next question, or do you still wanna get some time to catch up?
No, it's fine. I wrote it down already.
Mm-hmm. Okay. I'm gonna go to the next one. Which of the following processes include all others? Selective permeability. Oh, I hate that word. Movement of solute across the plasma membrane. Passive transport. Active transport. Movement of solute through carrier proteins. So what they are asking is what is the big topic here? What is the topic that covers other topic?
isn't it A?
How do you come to A?
Well, I picked A because doesn't isn't that like the starting point for the membrane before passive and active and the rest?
Mm-hmm.
So that's how I see it. Like that's how I get it. That's the starting point.
Hmm. Yeah, because everything fits under that one, right?
Yes.
Mm. This question isn't hard. It just requires memorization. So you're right. Next one. Hormones are stored inside membrane vesicle. So I'm gonna draw a long like decoding the question. Hormones stored inside the vesicle. So this is a cell. We have vesicle and we have hormones, happy hormones. These hormones can then be released from cell when the vesicle feeds with plasma membrane. So When it fused with plasma membrane, it will go out. This is an example of diffusion, pumping, phagocytosis, exocytosis, endocytosis.
Isn't it D?
And your reason?
Because exocytosis doesn't exo mean it's going out, and then endo isn't it going inward?
Mm-hmm. Yes. Exo out, endo in, right? What about phagocytosis?
Doesn't it gulf up?
Gulf of cell drink, right? It's like drinking stuff. What's not diffusion?
Oh, okay, I get it now. I'm sorry. It is diffusion because it's going across the plasma membrane.
Oh, I think you fall into my trap. You.
Oh, yeah. Oh no! It's exocytosis. Oh, never mind.
Yeah. I was
Okay,
just asking you to like reason everything, reason one by one. Yeah,
okay, yeah, it makes sense now. Oh my gosh, that's what happens.
that's fun. Okay, Samara, do you have any question about this one?
No.
Okay, and you have it down?
Yeah, I wrote it down.
Okay, next one. Why is it? Ah, damn it. Okay, so I already gave you an answer, but we're still gonna go over it. Okay, I'm sorry, guys. The difference between pinocytosis and receptor mediate endocytosis is that, first off, do you guys remember what is pinocytosis is?
I just remember pinocytosis as well from the definition. Like the only part I remember is that it goes up.
You know what? I made a mistake earlier. When you say phagocytosis earlier, phagocytosis is cell eating, cell eat.
Oh, okay, yeah, I got those two mixed up.
Pinocytosis is cell drink, right? So when we eat food. When we drink liquid. So pinocytosis is when you take up the liquid. You take the liquid up. Phagocytosis, you take the macromolecule up. And receptor mediate endocytosis. Okay. Do you remember what is endocytosis?
It's going in.
Uh-huh. It's going in, right? Something was going in a cell. And what is receptor mediate means?
I forgot that one.
What is receptor? Just asking you from like general understanding. What is receptor?
Doesn't it receive?
Receive, right? Just like when you got notification from your phone or your phone ring or something, you got signal in, right?
Right.
Mediated means depend. Mediate means depend. So it means that it depends on signal to take things up. Receptor mediate endocytosis. So how does cell drinking different from cell taking up food when signal comes in? Well, just me saying it, it's kind of like already descriptive of what's the difference, right? So let's look at a choice. A, pinocytosis bring only water molecule into the cell, but receptor-mediated endocytosis bring other molecule as well. Well, this is not really correct because like it's not only water. It's just everything that's liquid, everything that's like a small molecule can come in through pinocytosis. So we're gonna hang on for A. For letter B, pinocytosis increased the surface area of plasma membrane, whereas receptor-mediated endocytosis decreased the plasma of membrane surface area. Well, pinocytosis is actually the not both of them do not increase nor increase the surface area, all right? The surface area is always going to be the same. So this one definitely wrong. Pinocytosis is non-selective in the molecule it brings into the cell, whereas receptor-mediated endocytosis is selective, triggered by certain molecule that bind to receptor. So what they're saying is that pinocytosis take whatever in, which is liquid. It takes whatever in and constantly take it in. And receptor mediates take stuff in when there's receptor. So this one is definitely right. The pinocytosis cannot bring in water molecule from extracellular fluid, while receptor-mediated endocytosis can. We know that this is totally wrong because it can bring water molecule in. It can bring water in. It doesn't only bring water, it brings other stuff too, and non-selective. So this is wrong. E, pinocytosis has a weird name, while receptor-mediated endocytosis in just plain old confusing. Oh, this one is just straight out silly. All right, she will have one of those like answer choices like this. You can take it out right away. Okay. So, any question for this one?
So pinocytosis is not really picky with whatever comes in.
No, it's not picky. It's take whatever it has, but it takes it only like a fluid in a small amount. But for receptor mediate, is that it takes big things only when there's receptor coming in.
Okay, it's making sense now.
Okay, make sense? Okay. Should we go to the next one?
Yes.
Samara, you're okay?
Yes.
Okay. Okay. Answer the following. Is this exergonic or endergonic? Is this catabolic or anabolic? So I'll give you several minutes and let me go review my dinner. Hello. Any answer for me yet?
Is it endergonic, and anabolic.
Okay, define each term for me first. What is exergonic? What is endergonic? What is catabolic? What is anabolic?
So I know exergonic means it's going out, endergonic means it's going in.
Mm-hmm.
Catabolic, I know it's a destruction of large molecules. Sorry, hold on. My memory. I'm like not looking at notes right now. I'm trying to like just remember from my head.
No, you're doing good. You're doing real good.
I know that anabolic has to if it's I don't know if this is right. It's if it's positive, it's endergonic, and if it's catabolic, it's if it's negative, it's exergonic. That's kind of how I remembered it.
Okay, so you remember exer is like out, right? Ender is like in. So in these terms, like when you apply to like activation energy graph, it means exergonic means energy comes out.
So then,
Endergonic mhm.
I'm sorry, Rory. I'm sorry to interrupt.
Are you gonna say something? It's okay.
Um. Oh no. Okay, I got mixed up. Okay, so the purple lines where it says reactions without enzyme is it exergonic, which makes it catabolic?
Okay, so these two are the both purple and red line is the same thing, right? But it's just this one when you use enzyme, so it will lower your activation energy. Enzyme. But overall, these two are the same thing. So exergonic means energy comes out. Energonic means the energy got put in. And when we look at the graph, you see how it have to fight to go up to be we start lower and then you end up higher, right? So that means you have to take in energy to go from low to high. Right? It's how like you have to fight through life and put a lot of energy in order to end up in a higher position. Now, if the graph goes from high to low like this, now you see that you go from high to low, so the energy got come out from the graph. So the energy come out. So now do you understand? Is it exergonic or endergonic?
I understand it now.
Okay. So what is this? This is what?
So if it's going from high to low, it's exergonic?
And in this graph, is it high to low or low to high?
Oh, it's low to high. So isn't it endergonic?
Yes, endergonic. Now, is it catabolic or anabolic?
Isn't it anabolic because, well, from what I remember, that anabolic's usually endergonic?
Mm, no, you can think like that. Yeah. Okay, it's okay. Yeah, you can pair it like that too. But what I remember is that the cat like to destroy stuff. So if it destroys stuff, then you would go from it will destroy the big piece of molecule, and then you would go into like smaller. So if you destroy something, the energy will come out. But for anabolic, it's merging stuff together. That's what anabolic is. So from small stuff merged together to be a big one, and then you need to put energy in. I don't know if this is helping or not, but like, yeah. But cats are evil. They're gonna destroy stuff. Yeah.
So, oh yeah, because I know catabolic is the destruction of large molecules, and anabolic is the construction
Mhm.
of large molecules. Well, wouldn't it? So it's not anabolic?
Hmm?
Okay, so the answer is not anabolic, or it
The
is?
answer is anabolic. Yeah. I'm sorry. I didn't tell you the answer. Yes, you got the answer right, but I just want you to understand, really understand, like, yeah, everything about the definition. So yeah, however she asks about this, you can answer her. Yeah, depends on like. Okay, next one. Pepsin is a stomach enzyme that catalyzes the hydrolysis of protein, which our finding could increase the function of pepsin after you ate a giant steak. Okay, I'm gonna give you a couple minutes to figure this out, and yeah, tell me whenever you come up with a answer.
I think it's D, increasing the pH of your stomach to 7.
Okay. Can you walk me through how you come to that answer?
I thought about it because usually the pH is what affects the enzymes, which would increase the function of the pepsin.
Mm-hmm. Okay, you're on the right track. pH do affect how enzyme work, right? And specific enzyme work in specific pH. So now, do you know that our stomach does it work in neutral, basic, or an acidic pH?
I'm sorry, could you repeat that?
Our stomach, do you know if our stomach is basic or acidic?
It's acidic
It's
because
acidic,
it needs, yeah, to break down the food.
right? It's acidic. So if pH 7, is it neutral or acidic or basic?
It's neutral.
Mm-hmm. So you just said it, right? That it's working acidic. But
Yes.
if we increase to neutral, that wouldn't work. No, right?
What's it say?
Oh, yeah, I see now. Yeah.
Mm-hmm. So that would make us eliminate D. D is not right. Now, want to take another shot of answering?
Or is it E?
Hmm. Hmm. Looks very promising, and
Well,
I think it is. Yay. Yes. How do you come to that answer?
that was just a good guess, but how I did it was when an enzyme is being catalyzed, I know it increases something. So that's why I picked that it increased its overall concentration.
Mm-hmm. Okay. So there's a couple things you have to know about enzyme. Hmm. So one is that pH affect enzyme, right? Just like how we talk that no pH affect enzyme. Just how we talk about how in stomach it needs to be acidic in order for us to digest. Another factor is temperature. So we have to be in certain temperature in order to function for enzyme. But dropping your body temperature by eating steak, that's not how it works. Our enzyme in our body actually works at like the room temperature. Okay, so no, that's not a good choice for B. Doubling the activation energy needed for a reaction. Now remember from the graph that we see earlier, right? The graph of like endergonic. Now they ask you to increase this part to increase more energy more into this curve, so you would get like an even higher curve and you need to use more energy, which if you need to use more energy, then that's not going to increase the function, right? Because think about you yourself, if you have to put more energy to something, you wouldn't do it, right? So double activation energy, no, not it. C, inhibiting pepsin cofactor binding site. Do you guys remember what's a cofactor?
All I remember is that it's inorganic molecules.
Okay. And what's it? What is the function?
The function.
Function of cofactor.
Is it binding to enzymes?
Yes, it's bind to enzyme. So its function, just remember, basically it helps with for enzyme to function. It helps enzyme to function. Now, if we inhibiting the cofactor, so that if you inhibiting cofactor, that's mean it's not gonna help enzyme. So it could not increase in function. So it's no. So that's why we come to E. Answer E. Okay, good. Any questions?
I think we're good now. Yeah, I just got lucky with that one.
Okay. Yeah, try to use a process of elimination when you're doing a question if you're not sure about anything. I'm sure you guys already used that already. Next one. High concentration of amino acid inhibit pepsin from hydrolyzing. Wait. Huh? Okay. High concentration of amino acid inhibit pepsin from hydrolyzing protein by binding to an area separate from active site. Which of the following statement is not true? Which one is false? Okay, I'll give you guys some times.
Worry. I don't know if this is right, but
Hmm.
I feel like it's A
Why do you think it's A?
because because in this example it does not have like I don't know like I don't think it has feedback regulation which has negative feedback.
Mm, okay, let's decode um, together. Okay, let's decode the question together. High concentration of this amino acid. I don't know what it is. Let's name it A. Inhibits pepsin from hydrolyzing protein. So the pepsin job pepsin enzyme is to do something with this protein, right? That's how it's supposed to work. And this amino acid inhibits, right? Meaning that this enzyme A gonna stop the function of this the amino acid A. It's going to stop the functioning of this enzyme. And we further know that it's gonna bind to an area that's separate from active site. So we know that we have binding site, or we call it active site right here, and we have the other site in enzyme right here. So that's where the A is going to bind. That's what the question tells us, right? So from reading these, it's true that they didn't really state that it's not like they don't say it's negative feedback or not. So we leave that open for now. This is example of allosteric regulation. So this is exactly allosteric regulation. So this one is true. So we're gonna eliminate that first. This is example of non-competitive inhibition. Seems like it's true because competitive inhibitions means that it's going to compete for the active site. So both A and the protein gonna both compete to bind here at one place. Okay. And this is an example of catabolic reaction. So hydrolyzing. Hydro means water. Lysing means breakdown, right? So catabolic, we talk about it, cats like to break things down. So this is actually catabolic reaction. Now, this is example of amino acid as a substrate for pepsin. This is clearly stated in the question that amino acid inhibits. It's an inhibitor. Substrate is protein because substrate is the thing that binds to enzyme, right? In order for it to do something else, whatever it is. So that's substrate enzyme. So therefore, the answer is E. So even though it doesn't say that it's negative feedback, the thing is negative feedback operates with this kind of stuff too. Like they have the same mechanism. So we can't be sure, and that's why we have to go through one by one to eliminate everything. Mm-hmm. Does that make sense? Question? Does anything bother you?
It makes sense now, but I feel like I have to still study more on this.
Yeah. Go back to enzyme, right? Go study more about enzyme and then review this question again. And then I think it will make sense more when you go back to see picture of of the enzyme. Mm-hmm. Okay, can I move on to the next question?
Yes.
Actually, I have to leave. Can you email me the recording,
Okay,
Mari, please?
sure.
Thank you.
Bye, have a great one.
You too. Thank you for your patience.
Mm-hmm. Bye-bye.
Bye.
Okay, so it's just two of us now. um, okay, let's move on. molecule that function as an oxidizing agent, aka the electron acceptor in redox reaction. Do you have a good understanding about redox reaction, or should we
Kind of.
kinda?
So
Okay.
I don't know if this is right. Is it C?
Why do you think it's C?
Because oxidizing means it's gaining electrons.
Mm-hmm.
Oh no, isn't it okay? Isn't redox like okay? Never mind. Isn't it A because redox loses?
Oxidizing agent gains electron. Oxidizing, the stuff that really oxidize. Oxidizing means losing electron, right? Oxidation and reduction.
Oh yeah. Oh my gosh, I got them mixed up again.
Oxidation means lose electron. You lose it. Lose. Reduction means you gain. Oh my god. No, 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 no. Come back. Alright. You gain. And so they said oxidizing agent, molecule that function as an oxidizing agent in redox reaction. Oxidizing agent means the that create stuff that create oxidation. Stuff that create oxidation. So therefore, we would say it gains electron. Oh my god, the answer is just right here, to be honest. Electron acceptor. If it's electron acceptor, you just gain electron, and then you gain potential energy. It's because there's a potential energy in electron, right? So therefore, if you gain electron, you gain potential energy.
Oh okay. It
Mm-hmm.
makes sense now. It's because of the redox reaction. But if it was the other way around,
Okay. If they ask reducting agent or something, yeah. Reducting agent means electron provider, electron donor. Oxidizing agent, electron acceptor, reduction. Reducting agent, electron donor. Okay? So this one is just memorization only.
it makes sense now. I always get the oxidizing and reduction like mixed up.
Yeah, don't worry. I feel like everyone did. Yeah, it's really confusing. Okay, we're good. We're good to move on.
I'm good.
Okay. Also, if you want, you can leave your mic open. I don't mind. It's also up to you too.
I know it's because my family makes noise, so that's why I kept like putting it on mute.
It's okay. It's okay. Just leave it open. Okay. This one is photosynthesis. So yeah, this one is photosynthesis. So um, take note of this, right? And then once you learn synthesis, come back to this one. I would say, yeah, take
Okay.
let me know when you're finished taking notes.
I take a picture.
Okay, you retook it.
Yeah.
Okay, next one. By the end of a fast, long, continuous run from the bottom of Bundy Drive to our beautiful Bio2 classroom at the top of the mountain, you would likely be using blank in order to produce blank per glucose molecule. Okay. By looking at the question, what topic are we talking about? Do you know?
I forgot worry, but oh, I know we're talking about ATP and glucose. So wouldn't it be a
Oh, did you just answer?
yeah? Is it A?
Okay. So clearly, I think you know that it's cellular respiration, right?
Yeah, I forgot worry.
It's okay. So cellular respiration. And let's go back to our first slide that I have. Okay. So so look at cellular respiration. And how many ATP does glycolysis make?
It's not two, right?
It's two, right? Two
Oh,
ATP
okay,
for
yeah,
glycolysis. For a citric acid cycle, how many is it?
two.
For oxidative phosphorus, this one is supposed to be 32 to 36. It's not like 34. That's like, that's not the right number. Okay.
Yeah,
30.
that's why I picked
Mm-hmm.
that first answer. 30.
See, don't be too fast in picking answer. Try go through everything first, okay? So it
Okay.
seems that it seems right at first, so we're gonna keep it. Citric acid cycle, is it two? Right? We talk about it, it's two, right?
Yeah,
Lactic acid fermentation. You remember that you learned about this? Do you remember?
I remember her talking about it, but I forgot.
Yeah. So the thing is, when you go to like glycolysis, you make two ATP, right? But in your muscle, when like when you run and you're running out of breath, there's not oxygen enough for you to do the citric acid cycle in a mitochondria. So you would go through lactic acid fermentation in your muscle, which produce 2 ATP, right? So this seems right. Glycolysis 4? Not really. Photosynthesis? We're talking about human right now, so no. Now we have to come back and consider what, in the context of what are they talking about. So by the end of a fast, long, continuous run. So you are running, right? They're talking about you. You're running from the bottom of the hill to the mount. You would using blank in order to produce ATP per glucose molecule. And as I said before, when you're running, this is you running. Okay, when you're running, you don't have enough oxygen. You don't breathe enough oxygen to go through citric acid cycle. So you would go to lactic acid in your muscle and you produce 2 ATP, right? So that's why this choice, none A and B doesn't right, and it's C that's right. Make sense?
So, I mean, it's making sense now, but oh, it's asking how many ATP. Okay, it makes sense now. Yeah, I just Took a while. It makes sense.
Okay. And you understand that we're talking about if you are running, right? You are running, so then you don't have enough. So citric acid cycle take place in mitochondria, right?
Right.
But mitochondria needs to use oxygen in order to do like to do A and B, citric acid cycle and electron transport chain. Now, when you're running, that's your muscle needs to use the energy fast. So that's why they're going through lactic acid without. So this process doesn't use oxygen. Okay, and it's fast.
Oh, okay.
Hmm.
Yes. I remember talking about this now. It makes sense.
Okay, makes sense. Yay! Next one. Yay. Okay, next one. Assume a thylakoid is somehow punctured so that the this one is photosynthesis. You want to take photo of it?
Yes. Is this recorded, Worry?
Um, yes. Do you want to record the recording of the video?
Yes, or if not, could you send me like the file that we went through today?
Sure, sure. Okay, so you don't want to take a picture of this, right?
Oh, I
So,
just did.
okay. This one is also no for you because you haven't learned photosynthesis yet. Wait, so when is your exam again? If you guys haven't finished photosynthesis yet?
It's on Monday.
So you're not gonna have photosynthesis or you.
She said that she was only gonna do chapters 7 through 10.
Okay, so photosynthesis doesn't matter. Okay. Um, what is the function
I
of
think she
is it is it in?
she said that she was gonna try to finish chapter 10 on Friday, and then it was gonna be on photosynthesis. So I'm kind of like, okay.
I see. Well, you have all the files and yeah, you have a recording, so yes. Um, Rubisco, Rubisco. This one is also photosynthesis, right? Rubisco. Yeah. I'm sorry. Just a lot of photosynthesis. We can do this one. A researcher team is working on design of a new drug for treatment of lung cancer. To be most effective, this drug must be specifically enter cytoplasm of lung cells while not entering the cells of other tissue. Which of the following characteristics would likely enhance the specificity of this drug? Okay. Tell me when you have the answer. Okay.
Worry, I do, but I'm not sure. Is this from like cellular respiration?
No, 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 no. This is just like I don't know what topic is it from. Receptor?
Because I was gonna. I was gonna say that I think it's C. I mean, I picked C. I'm not sure.
It's kind of say, yeah, it looks good. The answer looks like pretty promising. And then why do you come to C? Why? Why do you pick that one?
Well, I just I'm just guessing. In my opinion, I just when I was reading A, B, and D, it just didn't sound right.
Okay. So when they say to be most effective, this drug must specifically ah no. Okay. This drugs can enter the lung cell, the cytoplasm of a lung cell, well not other tissue, right? So that means we have to find something that lung cell have, but the other cell doesn't have. Okay.
Ah, damn! I was almost close.
Similarity of drug molecule to other molecule normally transported lung cells. This one is kind of
I'm
fishy for me. It's like,
yeah, I'm having trouble.
I'll think about it. Okay, because reactive hydro. Okay, A is obviously not right because anything that's hydrophobic is just general. It's just like doesn't distinguish. It can go into any cell. Phospholipid composition of lung cell plasma, plasma membrane. Every cell have like the same phospholipid composition. Specific of lung of the drug molecule for binding to mm, similarity of the drug molecule to other molecule normally transfer. I think the choice is written wrong. It should write similarity of drug molecule to other molecule that imported into lung normally transport into lung cells. Yeah, yeah.
Oh, okay.
This one is kind of fishy. Yeah, just yeah, but that's the point of asking question is that what factor can distinguish lung cell from other type of cell? That's it. Celery stalk that are immersed in fresh water for several hours become stiff. Similar stalk left in a 0.15 molar salt solution becomes limp. From this, we can deduce that the fresh water. Okay, so this one is about osmosis and how like the water move. Um, the water move in a cell membrane to environment. Okay, so a general principle before we answer anything. So the water move from high water to low water. Okay, and then yeah, let me know when you have the answer for this one.
Okay. Worry.
Mm-hmm.
What I picked was that it's hypertonic.
Okay, what is hypertonic means?
Hypertonic. I just remember it as the concentration goes from high on the outside and then low on the inside.
High concentration, high on the outside, low on the inside, right?
Yeah,
Let me try to draw it out. So high concentration of solute, right?
right.
So as I said that it moves from high water to low water means um, low solute to high solute. So according to your definition, low solute inside, high solute inside, then isn't it supposed to water go out, right? Is that what you understand?
Well, yes.
Okay, that's how you understand it, right?
Yeah,
And also, so that's mean hypotonic, you understand that the water comes in, right?
yes.
Okay. So hypotonic waters come in, hypertonic waters comes out. Now, if we put it in fresh water, it will become stiff. It becomes stiff means that it was loaded with water inside. If you left in the salt water, it become limbs. It means that water have left the celery stalk, right? So stiff fresh water comes in. Salt water, the water comes out. So does it match with what you answer? Because you said if it's in fresh water, it will be the water comes out, and if it's in salt water, it will comes in.
Oh no, it doesn't match.
Okay, so what is the right answer?
Wouldn't it be B?
B. The first time you answer C, right?
Yeah.
Okay, let me see. I think it's yes. Yay! Okay, so stiff means that full of water. Yeah, limp mean it's the water left, so it becomes like limpy. You know how yeah yeah.
This answer, so it's technically saying so the fresh water goes from being good to bad?
No. It's just saying that um you've seen celery, right?
Yes.
So it means that if you put this in like fresh water with 0% salt, so that means the water gonna come in into the celery and it's gonna be fat with water in a fresh water. But if you put it in a salt water, right, the water just kind of comes out and it will become like a sad celery because the water comes out and it doesn't have any water left inside the cell. That's it. Does it make sense? Like you put it in inside it. Yeah, that's yeah.
It makes sense. I gotta review this again.
Okay. Right. But at least you remember it right about hypotonic and hypertonic. Yeah, I give you
Yeah.
credit. Yeah, I give you credit for that.
Thank you.
All right, next one. So what time are you gonna leave?
Until you're finished, actually.
Okay. So I think I'm gonna finish at 8.
I have a question, Maury.
Mm-hmm.
Before you start again, what I've been struggling with, like when it comes to like her exam, I noticed that I have trouble when it comes to her. Like one question could ask for like two different answers for just one question.
Ah. Ah.
That's
Right.
why I didn't do good on the first exam because that was one of my issues.
Hmm.
I was wondering like how did you get better at that? Because I know I heard Trotner say that you literally killed bio.
Oh, she say that? uh, oh.
That's why I'm asking you questions.
Oh. The thing about here's the thing. I do not have any technique. So if you were to ask me earlier, I would say for you to use Enki. Do you know Enki?
Oh yeah, I heard of that.
You heard of it? Okay. So do you know how to download Enki?
I downloaded it, but I don't know how to use it yet. I don't know if I
Okay.
did
I'll
it
send you
right.
a YouTube link of how to use it. All right. So here's what I do. From day one that I learn anything in her class, I put everything in her slides into the Enki right away, right? And I start reviewing from day one. Now, if you do this every day until the day near your exam, right? You would have a pretty solid you have a solid information in order you have solid information and you get everything accurately. So then when you answer the question that has several right answer, then you can spot it right away. So it's just use just basis of having a good foundation of knowledge is what makes you able to answer multiple question right. And also you see how I went through all this question. I always ask you to reason to back up your reason, right? I see that you seems to kind of like jump to conclusion. You jump to the answer because it sometimes feels right, sometimes you not sure. Well, sometimes it's okay to do that, but what would make you what would make you be a better person taking a test is you have to back up every reason for your choice. You eliminate it. You go choice by choice. Why is this not right? Why is this not right? And this will help you when answering with two or more answer, right? Let's say if we're going through this, what if B, A seems kind of right, B is not right, C is not right, D is, D is absolutely right, and A, you know by heart that A is absolutely right. So that's when you can answer two right question, right? No, two right answer in one question. Make sense?
Yes.
Yeah.
Yeah, I want to pass this exam. I want to pass like all her exams, even the final, hopefully.
Yeah, I think you have a good knowledge from what I can tell. It's just that you have to be you have to slow down, back up your reason, right? Think through the question, eliminate one by one. You say to yourself, why is it not right? Why is it right? And
Yeah, you're right.
mhm,
Because yeah, I've been doing that. I know that's not good.
yeah. So try to do it as I say. Try when just imagine that I sit with you in the exam, okay? And like I imagine that my voice is walking you through the question.
That makes sense.
hmm
Thank you.
hmm And for the next exam, for the next before preparing for next exam, you prepare for it from day one, okay? Just like from the first day, you prepare, and then
Yeah.
yeah, you keep like reviewing one by one, a little bit by a little bit, and then that will become your great habit too, like throughout college career. That's you're gonna to be like prepare when taking a test, so you don't want to cram yourself.
So when you would still do this, so you never read the book at all, like
No, 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 no. So
okay.
yeah,
I
remember
didn't-
I told you I think I used to tutor you for Bio 1. Am I right?
yeah,
Yeah.
yeah.
So for Bio 1 you need to read a book. Bio 2 don't need a book at all. You don't read.
Okay, because I've been doing that, and I do notice that I'm like wasting more time reading the book instead of actually learning the material.
Yeah, that's why I said to use Anki, because here's the thing. If you use Anki and you start reviewing right away, you do not waste time any one bit. You go straight to reviewing. And that's what we want to do. We save time. And also we're like a busy students too, right? We don't you have
Yeah,
responsibility, I have responsibility. We all have to do like other stuff too. And we all always tired. We always want to rest. So yes, listen to her lecture, put everything into Enki right away, start reviewing every day. Like it's okay if you want to review like a little bit, even if it's just a tiny bit of like Enki, just try to do it every day. And then I think you do good. I think you don't have any problem with remembering stuff. Sometimes
yeah.
sometimes you're lack in like here and there concept, but overall you seems good. You seem solid.
Well, that's good to hear because I'm kind of like I'm just like very like nervous for this exam.
hmm
Yeah,
Yeah.
because my grade went down. That's why I like started freaking out. My grade went down from like an A to an F, so that's why I'm like I'm close to getting a D, but still like I want to pass this class with at least a B. Like that's my goal.
Uh-huh. Yeah, I think you can do it.
Yeah, I
And also Kelsey Kelsey the person you have what do you call it? Supplemental session with?
yeah, but there's a problem. I did talk to Trotner. I can't make it during that time because I have OChem lab
Mm-hmm.
on Wednesdays.
Mm-hmm.
When she does it, she does it from 6 to 7, I think, for like an hour.
Mm-hmm.
but my OChem lab starts at 6:20, so I can't make it.
Mm-hmm.
And
I
I did
see.
email Kelsey about like if she did either like she can record it or I can meet with her, but I can't meet with her because she said that she's been working
Mm-hmm.
and she couldn't record it because supposedly there's I guess she didn't it didn't allow her to record or something went wrong.
Mm-hmm.
So I've been going to office hours with Trotner.
I see. Yeah.
And I was gonna ask you, so are you tutoring for now on for Bio2?
Yes, seems like it.
Okay, because I can't go to you. I mean, if our schedules are good, but I can't go to Kelsey because of OChem Lab.
Yeah. I was gonna ask like, yeah, if Kelsey go over solving some problems with you, cause I remember I heard from someone that Kelsey do not just go over the homework, which is helpful to some level, but when I was like last year, we have Arlene as our supplemental, and she would give us tons of problem, and that's a way that I learn and practice. It's doing problem, reviewing. So that's how I do it.
So did you do the problems from the back of the book and from like the website from the book?
No, I did it for Bio one, because we didn't have any supplemental right? We didn't have supplemental in Bio one.
We didn't until like this year. They did it.
They did have okay. Yeah, so that's how at first I started out with practicing like that. Um, but now for Bio 2 I practice based on what Arlene gave us, the practice problem, which is like very accurate of what's going to be on the exam too. It's not like all of it, but you can see like you can get a feel of it how Schottner like write her question.
Yeah, I learned my lesson. Yeah,
Yeah.
pretty much. Yeah. Yeah, I messed up on the exam one, but I feel pretty confident about exam two.
Mm-hmm.
I've been using, I don't know if you heard of this app, but I've been using Notion.
Is it on iPad, right? That app?
Yeah.
Okay. Let's do this one, last one, and then let's I'll let you go, okay? And
Yeah. Oh, sorry if I kept asking like
no, it's okay,
a
it's
lot of
okay.
questions.
I know that you really want to pass, and I really hope that you pass too. Yeah, that's why I'm here. It's okay, it's okay. Okay.
Thank you.
In which of the following environment would there be a greatest needs of osmo regulation? Osmo water, right? Regulation of water. Which environment would need regulation of water the most? And then tell me when you get the answer. Okay? Take your time.
Okay.
อันนี้2 ทุ่มแล้ว
Is it B? So
Mm-hmm.
I got B.
And how do you come to that answer?
Just by the hint that you put that it was water.
Mm-hmm.
That's all I remember. I forgot. I forgot the rest. Is this from? I have a question. What chapter is this from again?
Mm, osmosis?
Oh, okay. Oh, so okay. Oh, it makes sense.
Mm-hmm. To be honest, okay. If I were to walk through with you, let's read the first one. In animal, connective tissue cell bath in isotonic body fluid. Do you remember isotonic?
Yes.
What does it mean?
Um, all I remember is that it's like I think it's like no net movement of water.
Uh-huh, no net movement. Also, iso means same, right? So inside
Yes.
and outside the cell is the same, so there's no movement. Okay. So A, you can eliminate out. B, salmon moving from river into ocean. Okay. So when salmon move into river from ocean, we know ocean is salty, right? And river, it's a fresh water. And salmon moving from fresh water into salty water, so from no salt to salt, that's when the cell most likely going to die. So that's what, because the water coming out from the cell, right? That's why we need regulation for this the most. But also, let's look at C, a red blood cell surrounding by plasma. Red blood cells surrounding by plasma. So cell surrounded by plasma, and we know the plasma is consists of 70% water. And well, it's really not a big deal, but cells still have to regulate it water to some point, right? But it's not like very extreme. It's not like from fresh water to salty water. It's not like that. D. A plant being grown hydroponically in watery mixture of design nutrient. Well, this one also need water regulation too, cause like grown underwater, like plant cells and a lot of water outside and lower water inside. That's also need a lot of osmolar regulation too. But B is the one because it's like it's the most extreme case. Yeah,
It makes sense now because of osmosis.
yeah.
That makes
Okay.
sense.
Okay.
Thank you
So
so much.
no problem. How many questions have we gone through today? Let me see. Um,
I feel better now.
you feel better? Do you feel like more confident?
Yes, because oh my gosh, all month I was
Mhm.
just nervous.
Yeah, it's. I think it's because of.
It's because like I was freaking out because of my grade because that's the only bad grade I have.
Mm-hmm. I totally understand it. Yeah. When I get bad grades, I freak out too. So yeah, it's okay. It's okay. It's okay.
I just have one more question.
Mm-hmm.
I know I keep asking you, but like, she had posted like the outlines for the chapter, so just ignore that, right? So for me, just go through the slides.
Outline for a chapter.
Yeah, she posted the outline for chapter of the book.
She did.
Yeah. Do you want me to like send you like an example?
Oh, it's okay. I don't. It's okay. Um, I'll just focus on her lecture because that's what I did. Yeah.
Yeah. Yeah. Well, thank you so much for tutoring me and the advice. It really helped.
No problem. Yeah, because I think you need someone to guide you through, right? Yeah.
I do. That's why I kept asking a lot of questions.
I see a common trend here. Like people who come to me really try their hardest and also read the book too. So I think like reading the book is the pinnacle of downfall of losing your time. I feel like. Yeah. Yeah, you
Yeah,
really yeah.
it is. I agree.
Hmm. So use your time wisely and just focus on her. Yeah, on her lecture. That's it.
Okay.
Hmm.
Well, thank you.
No problem. All right. Have a good night, and then I'll email the video with um the slides for you for
Okay.
everyone.
Thank
Yeah.